Good morning, everyone. Thank you for coming to my talk. This session kicks off with my talk telling you how small inhibitors modulating CFTR's activity. So no conflict of interest. So we all know CFTR is a chloride channel. It uses ATP as energy to transport the chloride across the membrane. Lower activity resulted from either lower open probability or diminished conductance can lead to CF. However, this does not mean that the higher activity is better for CFTR, because if the activity is higher than physiological needs, it can also cause pathophysiological conditions such as secretory diarrhea and polycystic kidney disease. So for CF, we have developed many compounds to alleviate the symptoms. However, the drug design to curb CFTR's hyperactivity has not been well explored yet. So I graduated from a lab that studies CFTR's function using patch clamp, where I mastered the inside out and the wholesale patch clamp technique. So many times in my experiments, when I wanted to shut down CFTR, I would apply a compound called inhibitor 172. Here on the left, in the case of a lock open mutant, you can see the current is almost instantly shut down to baseline level by this compound. And on the right, in the wholesale experiment, even after the current is significantly decreased by a modification of a cysteine, so the application of this compound can still further inhibit the current to its almost baseline level. So this compound was first synthesized in Professor Alan Workman's lab. It's very potent, but for a very long time, no one knows where it binds on CFTR and how it works on CFTR. So also from here, you can see that this compound must be very hydrophobic and membrane permeate. So is it possible that it also binds at the protein-lipid interface, just like what we saw before with the CFTR potentiators and the correctors? So to answer all these questions, we decided to determine the cryo-EM structure of CFTR with the inhibitor 172 complex. So to do this, we collaborated with doctors John Capis and Wendy Young at the University of Alabama. They developed an expression system that uses a lentiviral vector that can overexpress large amount of CFTR. So for our purposes, we chose to use the E1371Q sample because this sample has a very good homogeneity due to the hydrolysis deficient feature. So the protein can be purified very nicely with the size exclusion chromatography with a very sharp and symmetric peak. And the purity of the samples are very good for both our control and our complex. And for single particle cryo-EM, a good monodispersity of the protein molecule is a must. So I checked the samples first with negative staining. At room temperature, you can see the particles separate very well from each other. And even after I put them on the cryoglaze, the particles still has very good monodispersity. This facilitates later particle picking and processing down the road. So for both our samples, the control and the complex, we got a very good final electron density map. For the control, it reached 3.4 angstroms overall resolution, and for the complex, it reached 3.6 angstroms. And here, from the local resolution, you can see majority part of those maps have very good resolution, which allows us to build very credible structures. So very importantly here, I compared our control structure with that previously published by Professor Jue Chen's lab, and you can see they are almost identical here. This suggests that the lentiviral expression system is equally good in producing good CFTR proteins for structural studies. And upon examination of our complex map, we found there is an extra density here in the pore of CFTR. It locates right below the narrowest part in the pore, which is here. now we know this is CFTR's gate and selectivity filter region. And the size of the density, corresponds well with that of the inhibitor. But when I tried to build the drug into the density, I found that the drug can be fit in there in two poses. So to find out which pose is more favored, so we turn to MD simulation. So in the MD simulation, you can see for the pose 1, in some experiments, the drug is not very stable. It moves away from the density. But with pose 2, the drug is very stable when it binds there. So we think that the pose 2 is much favored. So here is the side view and the top view of the drug in the pore. You can see here the interacting transmembrane segments are PMs 1, 6, 8, 9, and 12. And the drug interacts with CFTR mainly through hydrophobic interactions, also with a pi stacking effect. And especially there is a salt bridge between lysine 95 and the carboxylate of the inhibitor. And this salt bridge can be direct or water mediated as we measure the distance here. And this salt bridge is quite important as shown in our electrophysiology experiments. Here, for example, the 0.5 micromolar inhibitor can inhibit almost 80% of the wild-type CFTR current, but once this lysine is mutated, the inhibitor of the same concentration can probably only reduce 30 to 40% of the current. So as a matter of fact, that all the mutations we tested around the binding site decreased the potency of the inhibitor, as you can see from the right-shifted dose response curves. So now with the structure, we can answer the question that how this inhibitor inhibits CFTR. And as I told you that here is the narrowest section of the pore for CFTR. Most apparently, when it binds here, it blocks the ion flow through the pore. And on top of this, we found that it induces conformational change at the extracellular region of the transmembrane domain. Specifically, the extracellular half of TM12 moves into the pore, while that of TM1 and TM8 swing away from the pore. This leads CFTR into a closed state. So our previous functional study did predict that this inhibition involves a post-binding conformational change. And now we can see it with the cryo-EM. And we also noticed that different CFTR orthologs showed different sensitivities to this inhibitor. So we know the sensitivity of the channel or the potency of the inhibitor depends on both the binding and the post-binding conformational change. So with those mutations around the binding site, I just showed you, you have seen how changing binding can change the potency of the inhibitor. Then how does the post-binding conformational change determine those different sensitivities for different CFTR orthologs? So to look into this aspect, we noticed that pig CFTR, very interestingly, it conserves all the amino acids involved in the inhibitor binding. So those are the green ones labeled out. So, but surprisingly, pig CFTR is much less sensitive than human CFTR if you compare the black curve with the orange curve. So, but interestingly, if we put the transmembrane domain 2 of human into the pig CFTR, the sensitivity of the chimera channel becomes very similar to that of human CFTR. If you look at the blue curve and the black curve. On the other hand, if we put a pig CFTR, the TMD2 into human CFTR, the sensitivity of the chimera channel then becomes very similar to the pig CFTR. It's the green curve here versus the orange curve. And we didn't see much change if we swap TMD1 between these two orthologs. So here I must say that we don't have the structure of the pig CFTR bound with inhibitor 172. But if we can accept the presumption that pig CFTR also binds to the inhibitor through those same amino acids, then here these data tell us inhibitor 172 most likely inhibits CFTR mainly through TMD2. So as a matter of fact, if we only swapping TM8 between these two orthologs, we already still can see the sensitivity change for the chimeral channels. So from previous studies, we also know that TM8 is very important for CFTR's gating. And we also know that the famous CFTR potentiator VX770, aka EVA captor, also binds very close to TM8. So this made us wonder, how binding of those potentiators would affect the action of inhibitor 172. So we did the inhibition experiment in the presence of VX770, and we found that the IC50 curves shifted to the right, which means the potency of the inhibitor is impaired. If you look at where they bind on CFTR, you will see that they bind they bind almost to the same level to TM8, but on the exactly opposite side of TM8. So inhibitor 172 in the pore, the VX770 outside of the pore. So we propose that these two compete for TM8 for different states of CFTR. Inhibitor for the inhibited state, the potentiator for the activated state. So in other words, they are antagonists on CFTR. So with that, I come to the conclusions of my talk. So the take-home messages are inhibitor 172 binds in the pore of CFTR, close to the narrow region to block the ion flow. It interacts with CFTR through hydrophobic interaction, pi stacking, and a very important salt bridge. Inhibitor 172 induces conformational change at the extracellular region of the transmembrane domain, leading the channel into a closed state. It inhibits CFTR mainly through TMD2. If you look at those conformational changes, actually, they mostly happen in TMD2. So inhibitor 172 and the CFTR potentiators contact each other from the opposite side of the important TM8 to favor the inhibited or activated state. Here are my acknowledgements. For this project, I would like to thank all the members in our team and at EM Core of University of Missouri for their hard work. Here is a big shout out to our collaborators at the University of Alabama. So they did a very good job on protein purification. So their lentiviral expression system is so far the only alternative to the BACMAM expression system on large amounts of CFTR protein production. I also thank our collaborators from Sweden on the MD simulations. Here is a special acknowledgement to the staff and the facility at the National Center for Cryo-EM Access and Training. So I have been a continuous user of that facility for the past 8 years since I was a postdoc. So I received tremendous assistance and help from them. So a big thank you to the foundation, of course, because the foundation supported our research for the past 3 years. As a matter of fact, here the CFTR protein production core is also supported by the foundation. So it's again the foundation that makes this project possible and successful. So I also thank our other funding sources. Thank you very much for your attention. I can take my questions now. We do plan to study mouse CFTR. In the lab, currently, we have someone working on that construct, mouse CFTR construct. So once that is done, I'm pretty sure many experiments like the one you just said can be done with mouse CFTR. We are very looking forward to that construct.
Oh, mic's on now. So do you find it surprising that you can swap out domains of the pig CFTR with human and not like disrupt some secondary interaction? It's interesting, ferret is completely refractory to CFTR 172. Doesn't inhibit it at all. Ferret CFTR.
Yeah, I remember we did get a question from the reviewer about the ferret.
Wasn't me.
Oh,
Unless it was positive, then I'll take credit.
Did you just say was you or was not you?
No, it was
no.
not me.
Okay. So we did look into the sequence alignment between human CFTR and the ferret CFTR, but it turned out the ferret CFTR does not 100% conserve all those binding sites. So then, this gives you many possibilities. So there's no way to answer it specifically how it works on the ferret CFTR, right? If those binding residues are different, even one is different, probably you will need to be very conservative on your.
Very nice, very nice. So pig has a higher single channel conductance than human CFTR. So one interpretation of that result is that the pore is sort of wider, or another alternative would be that there would be sort of greater attraction of ions into the pore. So the fact that INH172 has greatly reduced inhibition of TCFTR, could that be interpreted as that the pore constriction is wider in pig CFTR compared to human CFTR?
So that's correct. Pig CFTR's field is higher than human CFTR. Theoretically, based on our theory, that probably it should be easier for the pig CFTR to get inhibited by the inhibitor, right? But actually what we see is exactly opposite. So I think it's because human CFTR and pig CFTR has different TMD2s. As I said, the inhibition, the binding site residues are one thing. There must be some post-binding conformational change after that. So I think it's something different in TMD2 between pig CFTR and human CFTR that leads to the different sensitivity. Otherwise, if everything else is equal, you should be able to see pig CFTR is better inhibited, right? It's the opposite way, but what we see it's not that. So, but I cannot give you a clear answer what leads to that, but I can go as far as that, I think.
Thank you very much.
You're welcome.
The structural overlays are really interesting, and I'm trying to wrap my head around the 770 and the inhibitor overlay. They look very similar. So is it reasonable to interpret this in terms of an allosteric transition where the molecules are pushing it towards different states, or is it the same state and just the inhibitor is blocking pore?
You mean blocking the binding of VX770 or what?
No. It's in the pore, right?
Yeah.
So it's just blocking conductance, or is it?
I think it does.
Yeah.
Because
so it's one conformational state, then, not multiple. Is that true? Is that a reasonable interpretation? Like 770 and the inhibitor are stabilizing the same structure. Is that true?
No.
Okay.
So what I said is inhibitor in the pore, it inhibits the channel. It leads to those conformational changes. This leads the channel into the inhibited or closed state. But for the VX770, so far from what we see from structural study is that if you use the E1371Q CFTR as your sample, even you add VX770, the conformational doesn't change.
Oh.
So.
But they can be bound at the same time.
Huh?
But they can bind at the same time.
I think so.
Wow.
No one did that. So okay. So for this one. For this one, I showed you is actually the superimposition of two different structures.
Yeah.
It's not a sample with both compounds.
Yeah. It looks so similar. That's really interesting.
Yeah. Well, the difference you see here actually is caused by inhibitor 172, because if you take away both compounds and you superimpose that with the only E1371Q structure, it will look like the pink one there. Because the VX770, so far from the structural studies, what we see, it doesn't change anything for the conformation of the CFTR. Possibly it's because they use the E1371Q as a sample, because that one always has NBD dimerized. That makes it harder for VX770 to change the conformations. But since you choose to use that one, we and Professor Jie Chen's structure already showed there's no big conformational change.
Thank you.
Yeah, thank you so much. Thank you.
Right. Good morning, everyone. First, I really appreciate the organizer for the opportunity to speak to you today. And so today we are going to talk about the extracellular loop of CFTR. It's really a topic that we don't talk much actually recently. And hopefully through the talk, I will give you some kind of idea, how important of this extracellular loop of CFTR. So nothing to disclose. So my previous work is really focused on the ER-luminal chaperone proteins, and we demonstrate that ER-luminal chaperone proteins or the quality mechanisms is really important to mediate CFTR through to the degradation pathway, and specifically one of the chaperone we identified is called DNAJB9, and can bind to the CFTR within its extracellular loop, and then mediate CFTR through the ubiquitin and proteasome-dependent degradation. And importantly, knockdown DNAJB9 actually able to increase the expression of wild type and the delta L508. And not long after my study, Trikafta was approved by FDA and to treat people with CF. And generally, Trikafta, you see already, is very effective in rescuing delta L508. And I was wondering, if knockdown DNAJB9 actually can further improve CFTR expression, because they are targeting different domains of CFTR. So here just give you an example of the rare CFTR mutant. One is the A455E CFTR. You can see that single treatment, either VX44561 or knockdown DNAJB9, can improve CFTR expression. And with two different methods, we can further increase the CFTR expression. And using the same method, we test other rare CFTR mutants, and they all show similar phenotype, more or less. So clearly, there are two things we can learn from this experiment. One is that targeting DNAJB9 is the potential target that we can pursue to improve CFTR expression. And second thing is that CFTR extracellular loop is important for CFTR expression. And today, we are going to focus only the extracellular loop of CFTR. The hypothesis is that CFTR extracellular loop, or ECL, plays an important role in CFTR expression. So to do that, usually when you start something new, you want to know what is it and how does it look like and what we already know about this, right? And so this is the human CFTR amino acid sequence. You already know it has 1,480 amino acid, and I downloaded it from Uniprot database. And the extracellular loop is really highlighted in yellow, and you can see there are 6 extracellular loop for human CFTR. And the shortest one is extracellular loop number 5. It has only 2 nucleotides, and the longest one is the extracellular loop number 4, has 39 amino acid. In total, all 6 extracellular loop account for 105 amino acid, roughly 7% of CFTR whole molecules. So how does they look like? And especially we are interested in extracellular loop number 4 because it is longest and it has some characteristic of amino acid that could potentially interact with DNAJB9. So today we're going to focus on ECL4. So this is the dephosphorylated ATP-free human CFTR, and we can easily find them, 1, 2, 3, 4, 5, 6. Unfortunately, for the ECL4, most of the part is missing. Only some residues, a few residues on the C terminal is there. We can see that. So what are we gonna do? Can we predict that? The answer is yes. Nowadays we can do our job nicely without AI, right? So here's the AI predict models, and so we use the AI platform called AlphaFold and predict this structure. And And it seems like extracellular loop is protruded from the CFTR body. And but the AI also said, the confidence is less than 50%. So, and also the AI cannot tell us, is it important for CFTR expression or not. So we turn to other tools to understand more about ECL4. So here is the ECL4 of human CFTR. As I said, it has 39 amino acids ranging from the amino acid 880 to amino acid 918. And this is hydrophobicity map. And you can see that the red one is indicative hydrophobic and blue is more hydrophilic. And you can see, on both sides of the ECL4 is more hydrophobic and the middle side, middle region is more hydrophilic. It's interesting that, when we compare the ECL4 between different species, this pattern actually is well conserved. So you can see, from mouse, ferret, rabbit, pig, human, they all have pretty good hydrophobicity on both sides, but the middle is more hydrophilic. Although the hydrophobicity pattern is well conserved, the amino acid is not so conserved. And the black star showing you the conserved one. And you can see many of them actually is not conserved in amino acid level. So I'm just curious, to satisfy my curiosity, I just calculate how much similarity between them. So it turns out that CFTR ECL4 has really low similarity between species, even much lower compared to whole CFTR molecules. So sometimes I'm joking on ECL4 and with my lab, it has a competition between CFTR domains for the diversity, ECL4 probably will win a medal. So, but other than these things, there's an important feature of the ECL4. It has two unlinked glycosylation sites, the asparagine A94 and asparagine 900. Because of these unlinked glycosylation, CFTR usually appear bigger on the protein gel. And you probably know if you ever express CFTR, and you will know that CFTR has a human-made sibling, that 901 flag tag CFTR version, and in which the 901 flag is inserted right after asparagine 900. And this insertion actually interrupts the glycosylation consensus sequence so that the second glycosylation is missing. So it ends up that, the 901 flag CFTR is smaller on the protein gel. So here, just give you the idea how it looks like on the protein gel. And on the left is the wild type, and you can see 901 flag for both band B and band C, they appear smaller compared to the original or wild type CFTR. Same for the delta L508, and especially after you treat with the CFTR modulator, they look smaller compared to the wild type. So in order to avoid the influence from the 901 flag in our study, we only use the wild type CFTR. So how do we know about the effect of the ECL4 on CFTR? So to answer that, we actually performed aspartic acid screening on CFTR ECL4. So basically, we mutated each amino acid on the ECL4 into aspartic acid and look at their expressions. So this project is majorly conducted by a talent technician in the lab, Sarah, and she was able to use site-direct mutagenesis to generate every single aspartic acid mutation or variant. And then she expressed these plasmid in the cells and run the Western, quantify it, and then summarize it. So this is just give you an example of it. So in each panel, she has empty vector, wild type, and then several other aspartic variant. Before I show you the integrate data, And just mention one more thing, there's the asparagine 900 aspartic variant. And you can see it's so much smaller because if you recall, asparagine 900 is the second glycosylation site, and mutated to asparagine certainly will cause the loss of the glycosylation site. So it appears that the CFTR is smaller. So here is the data. So x-axis is the order of the amino acid. The y-axis is the CFTR expression level. The right line, we take it as 100, is the wild type CFTR expression. So anyone above this red line suggests has higher CFTR expression, and anyone below the red line is the smaller, has a lower CFTR expression. So generally, if you just look at the image, you can see that majority of the ECL4 aspartic mutant has lower CFTR expression, and only small fractions of CFTR variants show higher CFTR expression. And the interesting thing is that mutation to the amino acid in the middle region, where the hydrophilic area has higher CFTR expression, suggesting that amino acid mutation to aspartate in these amino acid probably promote CFTR expression. And therefore, the original amino acid in this region probably important to mediate CFTR degradation. And also, for both sides of amino acid, when you mutate these amino acid into aspartic acid, you actually got lower CFTR expression, suggesting that when you mutate to aspartic acid, you lost of them, and suggesting they promote CFTR degradation, and therefore the original amino acid actually is important to maintain the CFTR stability. To validate this data, we select a few amino acids and mutate them into alanine and see if we see similar pattern. So we chose 4 amino acids, A95, A97, 902, and 903. And among them, A95, A97, and 903 has shown higher CFTR expression, and 902 shows lower CFTR expression. So how does it look like when we mutate them into alanine? So here is the Western blot, and generally the pattern remains except the histone A97. So when we mutate histone A97 to alanine, the CFTR expression is going down instead of going up. So suggesting that this histone A97 could be, the effect could be a charge-dependent manner. So lastly, we want to assess if this phenotype will be recapitulated in the context of delta L508 in the absence and presence of CFTR modulator. So we mutate these aspartic acid variant with additional delta L508 and then express them in the cells. And you can see that in the absence of the CFTR modulator, the level is going up compared to the without the mutation. And this phenomenon is more obvious when we treat them with CFTR modulators. This is really mimicking the effect of knocking down DNA JB9. So we are still processing the message from the data, but what we can tell is that CFTR ECL4 is really important in regulating CFTR expression, in which it was underestimated before, and also each residue of ECL4 could be different, have different role in CFTR expression, and also suggests that interrupting the CFTR ECL4 and lumino-chaperone interactions may further improve CFTR expression. And with that, I would like to thank people doing the work, especially my technician, Sarah, for her dedication on this project. And I also want to thank Jorge in the lab, participate in this project, but he's on the other side of the project, working on the chaperone. So he has a poster 350. If you are interested, please stop by. Also want to thank the collaborator, AP Naren and Kavisha, for their help, and the group people at IU and my mentors. And I also want to thank CR Foundation and Well Center funding for the support. Thank you for your attention.
Questions? Jeff?
Really interesting talk. So I was curious whether the mutations that you've made, whether you've attempted to actually feed those into something like alpha missense to see whether you can actually correlate the predicted pathogenicity with the inhibitory effects you're seeing of specific mutations.
You mean the alpha?
Missense?
Oh, alpha missense.
Yeah.
Okay. ECF4, actually, when I look at its CFTR2 database, I don't see much mutations there.
But just to predict those that are rescuing versus or improving versus those that are inhibiting, you could just plug those into alpha missense and see if you can actually predict for further mutagenesis. I was just curious if that's something you
yeah, we haven't done that.
I have a second question, which is it's intriguing with the aspartate mutants and especially this looping, as you point out, a site of chaperone interaction. Of course, HSP70s really dislike negative charges. So I'm wondering actually just trivially if
That's a good question.
explored whether you have the pull down.
Yeah, we haven't done that. Yeah, that's certainly something the direction we want to pursue. Yes, thanks.
Maybe a bit related. I wondered whether you know whether DNA JB9 plays a role in phenotypes.
Yeah, I think so, but we haven't tested. Yeah, good point. Yeah, thank you.
So very intriguing results. One note of caution, although AlphaFold is quite good at predicting the CFTR structure in general, it is totally wrong from about 887 to 930. So in that case, I think you actually do need the experimental structure, probably because there's an inverted phospholipid that's stabilizing this weird thing in TM8. So, but the results are certainly very intriguing, and I may not have followed this exactly right, but the impression I had is that the aspartate acid mutations in the F508del ETI corrected had a much stronger effect than they do in wild type, which would make sense. If that's true, do you know anything about the kinetics in that case? In terms of, it looks like you're just probably getting a lot of degradation of the F508del that's offset by the aspartic acid mutations. Is that how you interpret that?
So you asking whether in the delta F508 context?
Yeah, was my impression, do those mutations have a much stronger effect on F508del ETI expression than they do on?
You mean additional treatment with the
fold increase in BNC with 508del compared to wild type?
Okay. Yeah, we haven't.
Because it seemed, but in your graph, I thought it was like a 50% increase up to a 50% increase in wild type, but it looked like it was a
only marginal
fold increase in F508del.
Okay.
Is that correct, that they're much stronger in the F508del?
Yeah, we haven't really close look at them, but the amplitude, but yeah, it's increased, but I don't know how much actually compared to wild type in delta alpha.
Nothing about the kinetics of the degradation yet. I guess that's about to come. So,
degradation we haven't tested.
You haven't characterized the kinetics of that maturation process.
Yeah, we haven't.
Yeah. Okay.
Yeah.
Thanks.
Thank you.
Martina, do you want? Short question.
When you inhibit interaction with chaperone and you get basically more complex glycosylated when C-made, is it also more stable? Is the half-life longer, or does it degrade as quickly as when it's active?
Okay, that's good point. We haven't tested. Yeah. This is like the transfection was done like in 48 hours, so it's kind of homogeneous that level.
Thank you again.
Thank you.
Okay, next speaker is Dr. Celeste Reep from Stanford. Welcome, Celeste.
Hey everyone, my name is Celeste. I'm a postdoctoral fellow in the Capito Laboratory at Stanford University. And today I'm going to talk to you guys about how F508 delta is targeted for endolysosomal degradation in the presence of small molecule correctors. I have nothing to disclose. So, as you guys well know, CFTR is synthesized at the ER, where it is core glycosylated, then traffics to the Golgi, where it is complex glycosylated, and then makes its way to the plasma membrane, where it functions as a chloride ion channel, and then gets degraded through the endolysosomal system. And this, of course, leads it to have this characteristic immunoblot pattern in which you have a core form and a complex form. Now, the most common disease variant, F508 delta, gets destroyed by the ER-associated degradation system before it reaches the Golgi. So what happens is that it does not fold properly at the ER and then gets sent to the proteasome for degradation, and this leads it to be only found in the core form or the ER form. Now, the main question that has been the focus of the field for the last 20 or so years is, can we generate drugs that restore trafficking? And our most successful drug strategy relies on small molecule correctors, elexacaftor and tezacaftor, which, as you can see, uncorrected F508 delta does not have a is not particularly well structured, particularly in this NBD1 domain where the F508 delta mutation is found. But when we look at the corrected form, we get an almost wild type conformation and this beautiful 2.5 angstrom structure. Now, despite the amazing effects that ETI has had for the CF community, 15% of people with CF on modulators are weak responders. And you can actually see this in the original sweat test data from the Middleton et al. Study. This is the clinical trial. What you can see is that with sweat test, you have some people who have almost no response to the drugs. And keep in mind that these people were all F508 delta plus a minimal function mutation. So we have to ask the question about why are 1 out of 6 people with CF weak responders? These could be due to genetic modifiers, something to do with the uptake or metabolism of ETI, the pathophysiology of CF. And the question that concerns me in my research is, can we identify drug targets that can act orthogonally to correctors to help these people out? And what I'm going to do is I'm going to look at the cell machinery that interacts with CFTR in the presence of correctors and see if by tweaking this in some way, we can actually enhance the stability and folding of CFTR. So the way I do this is I perform CRISPR knockout strains for drug targets in the presence of elexacaftor and tezacaftor. So I have an amnion green F508 delta cell line that also expresses Cas9. I infect with a guide RNA, I induce CFTR expression, and then I use FACT sorting to select for the top, 5% GFP positive cells, and then compare that to an unsorted fraction, and then I sequence the guide RNAs and calculate gene enrichment and the like. And so this helps me identify both enhancers and suppressors of correctors. So enhancers are great because these are potential drug targets that can act additively with correctors, whereas suppressors can give us some idea of the genes that might lead to corrector resistance. This system employs an amnion green F508 delta reporter, which enables real-time quantification of CFTR degradation with flow cytometry. So we have this CFTR molecule that is N-terminally tagged with amnion green. And if we give ribosome inhibitors to the cell, CFTR degrades and the amnion green degrades with it. And so therefore we can come up with this decay curve and we can calculate things like half-life, which in my system is about 40 minutes for F508 delta. So when we look at the degradation kinetics of F508 delta, we can see that ET has this amazing effect on the stability of CFTR. However, this doesn't actually bring it up to wild type levels of stability, which is fascinating because when we look at the structural data of F508 delta with correctors, and this is coming from the cryo-EM paper with ETI, what we can see is that most of the atoms in the corrected form have the same position as wild type, except for a handful here in this NBD1 domain. So another thing that we noticed in our studies of looking at this is that F508 delta is twice as likely to be targeted for lysosomal degradation than wild type. So we did these degradation curves with wild type, and we added in a lysosome inhibitor called bafillamycin A, and we found that by 8 hours, 8% had gone through endolysosomal degradation. In contrast, when we add in elexacaftor and tezacaftor, we see that 20% is degraded through the lysosomal system by 8 hours. And this is further interesting because F508 delta without any correctors is not degraded through the lysosomal system at all. So the question that I ask is, what is the post-ER machinery that is targeting F508 delta for degradation? So we did CRISPR knockout sublibrary screens with corrected F508 delta, and we found 2 classes of enhancers. One class involved the ER-associated degradation system. The other class involved the endolysosomal system. So to just give you guys a brief overview, ERAD is the process in which, or it's the main protein quality control system in the cell. So as CFTR gets translated at the ER, it folds and it gets triaged and ubiquitinated by the action of E2s and E3s, dislocated from the ER membrane to the cytosolic proteasome. And RNF5 was the top E3 ligase in our screens with corrected F508 delta. You can see it here. One thing that is really important to note is that the corrected and uncorrected ERAD machinery that is targeting F508 delta is the same. We don't see a shift into a different ERAD pathway when we add in elexacaftor and tezacaftor. And what we found is that when we knock out RNF5, it is indeed additive with correctors. So knocking out RNF5 by itself has no impact on trafficking, but has this nice synergistic effect when we add it in with correctors. And so we can see an increase of band C of about 50%. The magnitude of the change in degradation kinetics is the same between corrected and uncorrected. And what we think is happening is that normally CFTR is synthesized in this B1 state, and it goes directly to ERAD. And then when we add in correctors, that shifts the folding equilibrium into these B2 and B3 states that enable it to escape the ER and go into its post-ER form. And all that RNF5 is doing is just increasing this B1 state. And you can learn more about these state diagrams as well as genome-wide screens with uncorrected F508 delta in my recent publication. So the second class of hits that we observed were endolysosomal genes. And to be clear, none of these genes came up in our screens with uncorrected F508 delta. And we saw lysosomal machinery and enzymes, late endocytic machinery, and also RFFL, which was an E3 ligase that was previously implicated in corrected F508 degradation at the PM. So this is relief. We're working the way it should. But the thing that really surprised us was this E3 ligase called MyLip. So what is MyLip? MyLip is actually known as the inducible degrader of the LDL receptor. So what happens is during periods of high cholesterol, oxysterols bind RXR and LXR transcription factors. They transcribe this E3 ligase, and this E3 ligase ubiquitinates LDLR, and then it gets degraded through the endolysosomal system. When you knock out MyLip, you have more LDLR, you can uptake more LDL, and therefore it increases your intracellular cholesterol levels. So it also incidentally increases the stability of corrected CFTR F508 delta. So when you look at these degradation curves, you can see that knocking out MyLip has no impact on uncorrected F508 delta, but when you knock out the gene and add in the correctors, you do see this nice substantive increase in the protein half-life. And you can see there's also a nice increase in the amount of band C. So the question is, of course, is why would knocking out this E3 ligase stabilize corrected F508 delta? And so our first hypothesis was that MyLip was actually directly ubiquitinating CFTR. And the prediction was that if you can overexpress MyLip, you would increase CFTR degradation. However, when we added in a drug, which is an LXR agonist, we did not see an increase in F508 degradation, despite having a twofold increase in the mRNA levels from MyLip. And this suggests that it may not be ubiquitinated by MyLip, which kind of goes with what we know of MyLip. So it is known to be very specific E3 ligase. It only ubiquitinates LDLR receptors, which are these string bean proteins with one transmembrane domain and a cytosolic domain. And CFTR, of course, has a much more complex structure, and more importantly, it does not contain the MyLip binding motif. So then that led us to another hypothesis, which is that increasing the cholesterol content disrupts the degradation of CFTR F508 delta through the endolysosomal system. And so the prediction of this model is that if you decrease the cholesterol in the media, it will end up destabilizing F508 delta. And so that is what we saw. So we cultured MyLip cells in lipid-depleted media, and we saw a complete abolition of the phenotype. So if you compare the red, which is what we were observing, this nice increase in CFTR stability, and we then culture these cells in a lipid-depleted media, it is indistinguishable from the control. And this suggests that increasing cholesterol stabilizes corrected CFTR F508 delta. Now, we tried the opposite experiment. So we cultured cells in statins in lipid-depleted media, and that actually decreases the stability of corrected F508 delta. And this is not a MyLip environment. This is just our normal wild-type cells. And this is very interesting because it suggests that cholesterol levels may be impacting the response to ETI, which is important, of course, because in our patient population has about a 40% lower levels in cholesterol levels in the blood. So just to sum up, CRISPR screens identified two types of enhancers for elexacaftor and tezacaftor. We saw ERAD genes like RNF5, and we also identified endolysosomal genes like MyLip and RFFL. And we also have shown that high cholesterol levels can show a decrease in endolysosomal degradation. With that, I'd like to acknowledge the multitude of labs that I've worked with over the years on this project, including the Capito Lab, Bassick Lab, Gittler Lab, Weiss-Corey Lab, BA Lab, Olsman Lab, Porteous Lab, and Corrett Lab, as well as my funding sources, and particularly to CFF, which really saved this project from death's door early in the spring through their unfunded R01 mechanism. With that, I'd like to say that the Capito Lab is recruiting specifically laboratory technicians and postdoctoral fellows because we can hire you directly. And I'd like to say to just meet me at poster 310 because I'd love to talk more.
Yeah, thank you so much for great talk. Any questions from audience?
Do you think that a possible competition mechanism between the trafficking of LDL receptor and CFTR may also be involved?
Yeah, that could be involved. That's definitely something that we're looking into. Hi.
Hi. Do you think the increased stability is stability of the CFTR protein in the cholesterol-rich membrane, or do you think it has, or do you know whether it has, the cholesterol levels have an impact on the endocytosis and recycling rates? So it's more cell biological versus maybe biophysical.
Yeah, so this is interesting. So I just got this recent piece of data, and what I'm seeing is that actually MyLip has an increased level of endocytosis. than control, and that is a little bit backwards from what you would expect. So I don't know if it's like a competition thing, like, we're sort of, by increasing one form of endocytosis, we're decreasing the other. So yeah, there's still a lot of mechanistic questions that we really need to fill in for this. Yeah.
And another question from me. If you inhibit the proteasome, then it's very difficult to get complete, or even impossible, to get complete inhibition of degradation of F508del from the ER. So I wondered whether you have any hits that show that maybe there's ER phagy or autophagy already of the B band of the ER form?
Yeah, so we did not see anything with autophagy. And if anything, we actually saw most of the autophagy factors in the suppressors, but never in the enhancers. Yeah.
Those are very elegant studies. This is probably too much to hope for, but I'm wondering whether you've looked whether the cholesterol levels are actually increased with your knockdown, and if so, what cholesterol species are going up and down.
So
and similarly, whether you looked at whether phosphatidylserine knockdown might affect this.
Yeah, so we haven't done that. So the plan is that we're going to be looking where we take our lipid depleted cells and then just add back different derivatives, because right now we don't have a sense of like which specific cholesterol.
Although technically it's very hard to do that with the esters.
Yeah, it is going to be very hard. Everyone has warned me.
Fascinating talk. This is probably a naive question, but I'm curious, in the CF population, like your cholesterol metabolism problems, like CF patients on statins or other things, like, is there any correlation between the effectiveness of like Trikafta and that status of the CF patient?
Yeah, we have thought about this. We have not actually actively looked at this yet. Yeah.
I have a actual question. So
yeah,
great talk. And then this two pathway might be important for cell biology for maintaining cell homeostasis. Is there any toxic or harmful event happening in the cells or like tissues?
Yeah, so I know
when you knock out these pathways.
Okay, so RNF5 I think has actually been pretty well studied at this point to not have really many harmful side effects. So I think, of course, the Pedamonte group has been working a lot to develop inhibitors for RNF5. For MyLip, I think with this, we can mess around with cholesterol in ways that aren't necessarily going to be harmful to cell biology, because we could kind of do that with diet and stuff like that. So yeah, I don't know if that answers your question clearly, but yeah, we don't see a growth phenotype with MyLip or with RNF5.
Mm-hmm.
Yeah.
Thank you. Any other questions? No questions.
Okay. All right. Thank you.
Thanks so much.
So the next speaker is John Engelhardt, and the title of his talk, Pulmonary Ionocytes Regulate Chloride Secretion and Absorption via CFTR.
As you can tell, I'm not Fong Wong. I subtract 35 years, and we look amazingly similar. These are my disclosures. So I'm going to be talking about some recent studies that we've performed on ionocytes to try to get at some controversy that has been in the field recently. And I like to start typically when I'm talking about cell biology in the lung and function of epithelial cell types, to put it in context of differences between rodents and larger species. And the reason is that there's some biology in the background that at least spurred our interest in trying to understand the function of ionocytes. So in the proximal airway, in humans and larger species, these are the cell types that I have listed here. In the cartilaginous airways, goblet ionocytes, club cells are confined to the distal airways, the non-cartilaginous regions. And these are the cell types that express CFTR in the airways, in the conducting airways, that is. And ionocytes compose about 1% of the epithelium, but in the proximal cartilaginous airways, they express on the surface about 50% of CFTR mRNA. And there are some interesting differences between rodents and, or let's pick mice, humans and ferrets, I'm going to be talking about, that are different between where these regions where ionocytes and goblet cells exist versus club cells. And in mice, there's no cartilage within the intralobar airways of a mouse. They're more representative of the distal bronchioles in human. And ionocytes track with where submucosal glands exist. And these submucosal glands only exist in the proximal region of the trachea. So some work done by two Nature papers that came out in 2018, Aaron Jaffe and Jay Rajagopal, described manipulating ionocyte abundance in a number of different ways in two different species. And one key factor is in multiple organs, FOXI1, which is the transcription factors required for the specification of ionocytes. And in the airway, this can come from basal cells. If you knock out FOXI1, you get rid of ionocytes completely. And so in mouse, they knocked out FOXI1. They'd harvest tracheal airway epithelial cells, polarize them, and they saw a decreased mRNA, about 87%, but an increased cyclic AMP-mediated current. And if one overexpresses CFTR, this was done using ferret in this Nature paper, one sees an increase in CFTR mRNA and an increase in cyclic AMP-mediated chloride current. In a later study that we did, you could also manipulate ionocytes by enhancing sonic hedgehog signaling. Number goes up, increased current, increased CFTR mRNA. And in Aaron Jaffe's paper in human cells, they inhibited Notch signaling, which decreases ionocytes, and they saw a correlation with decreased current. So about 5 years ago, we were intrigued by these species differences when we connected up with Jay to collaborate. And part of, sorry about that. And part of the hypothesis is that this could explain differences between why CF ferrets, CF pigs, and humans get cystic fibrosis, but mice don't. And just to point out that submucosal glands are an important component of this, Welsh Lab, David Stoltz have really done some nice work in CF pig on that. So the hypothesis was that species-specific differences in the abundance of submucosal glands and their associated ionocytes might be responsible, and we really needed to understand what ionocytes were doing. So Feng Wang, who's giving a talk in an adjacent session, that's why I'm speaking for him here, and Grace Grazer, who's now an MSTP student at Iowa, embarked on trying to understand what ionocytes were important for. First thing they did was reproduce what was done in mice. We knocked out FOXI1 within ferret, and you can see here an on-phos staining of ionocytes with cilia here in red. You see this ionocyte marker, ATP6V. You can see these infrequent ionocytes in wild type, not in the knockout. These are just sections below. And if one takes the airway epithelial cells from these ferrets, polarizes them, and looks in chloride selective symmetrical chloride, no bicarbonate, and vice versa, symmetrical bicarbonate, no chloride, one sees that there's about a 67% decrease in both chloride current and bicarbonate current. Similarly, we were intrigued by the fact that epithelia for FOXI1 knockout, as well as CFTR knockout from ferret, if one challenge was small fluid and then looks 24 hours later, the epithelium become dehydrated. And here I'm marking the surface airway height, or that fluid, by this yellow line compared to wild type. So ferret pulmonary ionocytes influenced anion current where that wasn't the case in mouse polarized airway epithelia. And indirectly, this implicates the fact that airway epithelia dehydrate indirectly implicates that ionocytes are important for fluid secretion if they're driving chloride secretion as well. And so a number of different things. I'm not going to go into extraordinary detail, but I wanted to show this one slide that goes in a little bit more depth about how ionocytes regulate airway surface fluid, because I think it embarks and connects some discordance in the literature between a number of different studies. So we identified a number of years ago that CF airway epithelia actually absorb fluid more slowly than wild-type airway epithelia. It's buried in the red journal, which I think probably very few people have seen. If one compares FOXI1 knockout polarized airway epithelia with wild-type, one sees here that the FOXI1 knockout, this is following a small volume of fluid added to the apical surface of polarized ALI culture. One sees they absorb fluid more slowly than wild-type. This looks identical to a CFTR knockout. And if one even looks more closely and adds a very small volume to the apical surface of wild-type FOXI1 knockout that don't have ionocytes or CFTR1 knockout, what you see is that wild-type epithelia very rapidly absorb fluid. This is 1 hour following 20 microliters on a 1 centimeter squared transwell. And they actually become dehydrated. They absorb so fast. And we're looking at ASL height here on the y-axis. Then they rehydrate, and then they overhydrate a little. And it's a little, if you carry this out at a very defined time course, it's almost sinusoidal until it gets to an equilibrated ASL height. If one looks at the CFTR knockout, FOXI1 knockout, you lose both the absorptive phase, the acute rapid absorptive phase, and the secretory phase. And it looks sort of like over here where I've mapped the absorptive, overshoot, secretory, and then get to a homeostatic ASL height. And in FOXI1 knockout and CFTR knockout, that's lost. So in the absence of ionocytes or CFTR, ferret airway epithelia have defects in both fluid absorption and fluid secretion, at least using this assay. And so I'm not going to talk about a number of other things that we compare between FOXI1 knockout and CFTR knockout and wild type, but I've shown you some of this. They dehydrate, they have reduced anion current, they fail to alkalinize, so they are secreting bicarbonate and fail to alkalinize following CFTR stimulation, increased viscosity, impaired fluid absorption, and in vivo, FOXI1 knockout have impaired MCC if you look by PET-CT imaging spraying radioactivity into the lungs. So the lack of CFTR and ionocytes also leads to failure to take up anions directly into ionocytes. And I'm not showing this, but it was in the Nature paper using an anion sensor from Alan Berkman. And if you take out CFTR out of ionocytes, you also have reduced MCC. And so about the time that we were publishing our studies from the Nature paper, a number of other papers came out. And a lot of them disagree with each other. Some suggest that ionocytes are just involved in absorption. Some don't really look at all components, and some focus on various aspects. But one thing that was different between these studies include the model system, a lot of times the culture system, the approach to manipulate ionocyte abundance, which could be knocking out FOXI1 with Cas9 or overexpressing FOXI1. And so we embarked to try to compare. We hadn't really looked directly using asymmetrical buffers for absorptive currents through ionocytes. And so we started to compare to some of these studies. And John Hanrahan had a very nice study looking where they produced a chloride gradient where there's low chloride on the basolateral side of a polarized airway epithelium, high chloride on the apical side. And we had published in symmetrical currents that showed this data. If you knock out FOXI1, you lose about 60% or so of your current. And when we did the reverse gradient with gluconate on the basolateral side, indeed, this, whoops, sorry about that, this very, very large negative current, because chloride's going from apical to basolateral, is completely lost in FOXI1 knockout ferret airway epithelia. And you can quantify this, very significant differences between groups. And so, ferret pulmonary ionocytes facilitate both absorption and secretion. We feel the ionocytes' functional controversy is really caused by these differences within model systems, we think, and culture systems. And it's unclear the extent to which, if you take out ionocytes, that there are non-CFTR components of the ionocyte that are influencing this entire process. So what we felt and what Fong embarked on was a conditional genetic approach where specifically using pre-drivers and conditional CFTR ferret line, we knock out CFTR only in ionocytes and then ask secretory and absorptive questions. Is CFTR in the ionocyte responsible for that biology? And so there are 3 transgenic backgrounds in ferrets that we generated that were needed for this. The first, and these are actually described in the Nature paper, but we didn't do really in-depth studies on absorption in there. 2 of them are this driver or this Cree reporter that switches from EGFP or tomato to EGFP when you express Cree, and then a Cree driver that's embedded within the 3' untranslated FOXI1 gene, and you can lineage trace ionocytes here on the airway surface, and if you have the conditional allele, you'll delete CFTR only in ionocytes that were activated by induction with tamoxifen. So how does this perform? So here I'm just schematically showing a wild type epithelium in blue, CF in red. If you're looking at the wild type here, you see these apical caps of ionocytes using a BSND as a marker for ionocytes. If you treat, this is in vivo, so this is a ferret treated with tamoxifen, triple transgenic ferret, you'll knock out CFTR only in ionocytes. And here on the surface of this trachea, you can see the CFTR expression and apical caps are gone. And it's interesting, these apical caps are marked here in white, but we know by single cell sequencing, not all ionocytes express CFTR, and you can see one right here with an apical cap that lacks it. And we really don't understand the biology behind what's the difference between these type of ionocytes. Okay, so let's take this system into an ALI culture, and I'm calling here CFTR conditional knockout. That is CFTR knocked out only in ionocytes. So, and so this is wild type epithelium. We're starting a symmetrical gradient, low basolateral chloride. What do you see when you shift to this low? We're using DIDs to inhibit non-CFTR chloride channels. Shift to the low gradient, you see a pretty large current. You activate with forskolin IBMX, goes up, and here is predominantly the CFTR, the decline of GlyH101 that's inhibiting CFTR is CFTR-dependent current. And if you compare this to CF, this is now G551D, but no ivacaftor, BX770, so it's basically mutant. And comparing it to the conditional knockout of CFTR only in ionocytes, you lose completely all absorption of chloride in this negative gradient. You can quantify that here. And this is the GlyH101 response, very significant changes. So CFTR is required for the ability of pulmonary ionocytes to absorb chloride, and thus fluid, because fluid, if sodium's passing across the epithelium, where salt goes, fluid goes. So here is the alternative, and this was an experiment that we hadn't found in the literature yet, but one would predict, we had studied symmetrical chloride, we see reasonable gradients on the order of about 75 or so. So in this particular case, what Fong did was to start in low symmetrical chloride on both sides, and then move to high symmetrical chloride on the apical side. So what would you anticipate this to do? If ionocytes are secreting via CFTR chloride, you'd expect that current would go up. And indeed it does. It's about double what you see, including GlyH1 response as well as forskolin IBMX. And if you take out CFTR completely with G551D mutant, it's basically flatline, and this is the conditional CFTR knockout in ionocytes, where previously we had assigned about 60 to 70% of the current in symmetrical buffer systems is assigned to ionocytes if you use FOXI1 knockout. Here it's about the same, and this residual current is likely CFTR expression in secretory cells. And you can quantify this, and it's very significant. The GlyH1 response as well as cyclo-KMP. So CFTR is required for the ability of pulmonary ionocytes to secrete chloride and thus also secrete fluid. Now we took this one step further where we did short circuit current directly on the trachea of animals that were induced with tamoxifen to knock out CFTR only in ionocytes. And what we're showing here, here's a wild type. This is symmetrical chloride. It's really not feasible with a very thick tissue like this to be swapping, making gradients. And we believe, as I was taught by Bob Bridges, symmetrical chloride's the most relevant to in vivo. But so here we have wild type. You can see the CFTR mediated current. The averages are over here for GLIAD101. It's like a KMP inducible. And CFTR knockout only in ionocytes reduces it about 60 to 70%. So symmetrical chloride-containing buffers are most physiologic in my opinion, and it appears that pulmonary ionocytes in the ferret secrete chloride under homeostatic conditions. So this is no gradient. It's just asking what's the direction of the current under clamp conditions. So what's the physiological relevance of this? Does CFTR in pulmonary ionocytes inhibit MCC? And this was in the Nature paper, but I just want to show it here. So these are animals that were first, they're double transgenic animals that have FOXI1 Cre and conditional CFTR, floxed exon 16. Those animals were measured for baseline MCC. You can see here the radioactivity moving up the trachea. Then they were induced and immediately measured for MCC again, and very significant decline in clearance. So we believe that pulmonary ionocytes have functional roles in both fluid absorption and secretion. Ionocytes mediate fluid movement across the airway epithelium by both absorbing as well as secreting. And where salt goes, so does fluid. And ionocytes appear to be a gateway to rapidly move chloride under activating conditions. Doesn't mean it's the only way to move chloride. And even when you compare the CFTR knockout, the absorption, for example, between CFTR knockout, FOXI1 knockout, it's slower, but it eventually dehydrates. So there are multiple pathways likely to regulate fluid homeostasis on the airway surface, and ionocytes, in our opinion, appear to be the acute regulators of that, probably linked to submucosal gland secretion that's throwing a boatload of fluid out onto the airway surface and has to be rapidly equilibrated to mediate clearance. So ionocytes mediate almost, at least in culture conditions, mediate almost all the, or CFTR within ionocytes, all the absorption of chloride. I think that's probably consistent across most of the papers that have published on this, but are mediating a large fraction, but not all of chloride secretion. And I think it's important to point out that, I mean, ionocytes are limited in the proximal conducting airways. They're associated with submucosal glands. So what I've talked about is really relevant only to the proximal airways. And in the distal airways, Kenichi will tell you, I mean, there are other processes, other cell types that are performing these functions in the absence of CFTR. So I'd like to thank the group. Much of this work was done by Fong, at least the recent studies, and Grace Grazer early on, but it really is a team effort because working with a larger animal takes a village. Thank you.
Thanks, John. Great talk. Questions?
Yeah, I'm here from the same. So, I have two questions. One is, after you look the FOXG1 or the CFTR, you test another channel, for example, chloride channel in the membranes, the SLC, 6, 8, 9, so maybe, so maybe another channel, so you have some change effect.
Oh, when you knock, yeah, so when you knock out CFTR, do channels in other cells in the ionocyte change? Yeah. Yeah, we in the Nature paper, we did look at in FOXI1 knockout. So if you don't have any ionocytes, a lot of the ionocyte-enriched channels go up in other cells, including secretory cells. So there's definitely compensation going on, and it's probably important, you know, for CF patients how they deal with the lack of CFTR.
Because your function is you use ranking, so I just want to maybe another channel, so after you work out the result.
Yeah, I think it's a super important question. And I mean, what we're interested in are how the channels, not just in the ionocytes, but at our epithelial that touch them, how they coordinate this so elegantly. And what happens when you knock out CFTR, I think is an important part of the equation, because those channels that are modulated either up or down are likely important in the physiologic process when CFTR is there.
Ineka.
Very naive question. What is the mechanism of this absorption?
Yeah, so ionocytes are enriched in 2 of the 3 channels of ENaC. So we think it's probably the absorption is cell autonomous. I mean, everything says that it is from all the studies that are out there and published. The secretion's a bit harder to nail down, whether or not there are cooperative functions across different cell types that mediate that in coordination with ionocytes through gap junctions. But they have all the ionocytes are enriched at the single cell level, at least by mRNA, and the ones that we've localized are enriched in channels that are important in moving salt across the epithelium in both directions. Now, whether or not those channels are in the same ionocyte and it changes a state in order to adapt to the environment, I don't know. Or there are different subtypes of ionocytes, but it's not clear cut that these are absorptive and these are secretory.
Is it then all whether CFTR interactions directly play a role, or is it this indirect effect?
I mean, what's been studied about ENaC, CFTR inhibiting ENaC and all this stuff, I don't know if that pertains down at the cellular level to ionocytes like it does in other cells that it's been studied. Probably, I would say, but yeah, thanks.
One last question.
Hi, I'm really used to seeing ASL heights being measured in human cells. I was wondering if ferrets and CF ferrets showed the same sort of ASL heights as in human cells.
Yeah, in our hands, using the same culture media, I mean, the equilibrated ASL height in a human and ferret is somewhere around 12 to 15 microns. It's probably higher than it is in vivo, but that's the model system. But we don't see a huge difference. Yeah.
Thank you very much, John.
Thank you.
Yes. Our last speaker is Dr. Nadia Amin from Yale. Welcome, Amin.
Thank you so much for the organizers for giving me this opportunity to talk about the CFTR expressors in the intestine. I have nothing to disclose. So glad to follow John. In the early 90s, around the same time that John identified the pulmonary ionocytes, I identified cells in the proximal small intestine, both in human and in the rat, first by immunofluorescence. And these cells, as you can see on the left, have very high levels of CFTR protein in the apical domain, but also in the cytoplasm. They're predominantly on the villi, but they're also present in the superficial crypts. And we went on to do EM studies which confirmed that these cells were enterocytes, were electron dense, and about 50% of the CFTR was on the plasma membrane and 50% in intracellular vesicles, endocytic vesicles, recycling vesicles, consistent with trafficking. In the superficial crypt, the cells are enriched with NKCC1 in the basolateral membrane, supporting a role for chloride secretion from these cells. So the cells on the villi have a secretory phenotype. The brush border is very short and disordered. We also show that they lack absorptive hydrolases, including sucrase and isomaltase. They lack absorptive ion transporters, including NHE3, which is shown in red but absent in the high expressors. Very high CFTR, and they lack an important myosin 1A, which is important in development of the brush border in villus enterocytes. And we also show that this myosin was important for trafficking of CFTR in the villus enterocytes. But despite the absence of myosin 1A, these cells can traffic CFTR robustly to the brush border following second messenger agonists. So the myosin, or the motor that's responsible for the trafficking of CFTR in these cells, has remained unknown. We had a limited ability to study these cells over the years because of loss of tools, but in 2021, The Cleavers group was the first to use single-cell RNA-seq to demonstrate significant heterogeneity of cells in the human intestine. And in that study, he identified that cheese in the human duodenum enriched for bestrophin 4, which is a chloride bicarbonate transporter identified in retinal epithelial cells. These cells were also highly enriched for GUCA2B, which is the gene that encodes uroguanilin, which is a very important intestinal hormone. And Lane Clark has shown in the past that uroguanilin is very important in regulating cyclic GMP-dependent bicarbonate secretion in the small intestine and acidic portions of the gut in response to low pH. So since this paper, there's been several papers looking at BEST4 positive cells in the gut. These cells have been identified in a number of species, but not in mice. And that's similar to what we found in the cheese cells. They are not present in mice. But these BEST4 positive cells share a lot of gene profiles with the cheese cells in the small intestine, including BEST4, autopterin 2, which is a proton selective proton channel, carbonic anhydrases, and GUCA2B. But of note, the CFTR is only in the small intestine, and the BEST4 is throughout the gut. And the studies of the specification of the BEST4 cells in the colon suggest that they're regulated by NOT, like ionocytes, but they come from absorptive progenitors. So we've been doing some work with a geneticist at Yale in collaboration to try to understand the specification of the cells in the rat. And in addition to what I've published, what we are finding is that the cheese cells are regulated by NOT, but they appear to come from secretory progenitors. And importantly, they share a secretory progenitor with tough cells and enteroendocrine cells. And we currently have a paper under review on this. So what is the relevance of cheese to CF intestinal disease? Little is known, but what we do know is that the CF intestine is more acidic than normal, and the pH runs around 5. And the high acidity in the CF intestine contributes to the thickened mucus, the obstruction, the overgrowth, inflammation, malnutrition, and a higher risk for cancer. And the pathogenicity of all of this is not completely understood. There are several factors that favor these cells as being responsive to low pH and also in acid sensing. First, the location of these cells are in the proximal small intestine, in the duodenum and jejunum. Second of all, they are predominantly on the villi, so they face the lumen, where all of the CFTR is accumulated. Thirdly, the single-cell data, both in human and in rat, our unpublished data, indicate that these cells are enriched with genes that sense acid and respond through the cyclic GMP bicarbonate secretion machinery. And they're enriched for GCC, which is the receptor for uroguanilin, the hormone that regulates bicarb secretion, or TOP2, which I mentioned to you before, which is the proton selective channel, the acid sensing receptor GPR4 and BEST4. But in contrast to pulmonary ionocytes, we found, and I'm not going to show you the data, that the cheese or the intestinal epithelium period does not express FOXI1. Instead, cheese are enriched for a homeobox gene, MICE1, and we confirmed that the protein level shown here, that MICE1 is confined to the nuclei of cheese, both on the villi and in the superficial crypt. And so we can use antibodies against MICE1 to mark these cells in our double labeling experiments. And we did just that. So we looked at the single cell data and we found a number of proteins that are important in traffic and cyclic GMP activated bicarbonate secretion and pH sensing. So I showed you these two panels, B and C, before, but we found that the cells are enriched for myosin 1B, and you can see the co-localization of CFTR and myosin 1B in the apical domain of these cheese cells. BEST4 is enriched in the basal domain of these cells and extends into basal processes in the lamina propria. And the cells are also enriched for tubulin, part of the microtubule machinery that is important in trafficking, which we know the cells do. Like I said, they're enriched for uroguanilin, which is the intestinal hormone that is very important in regulating bicarbonate secretion. And the cell is chock full of uroguanilin throughout this apical domain and also extending into the basal processes. Similar, GCC is highly enriched in these cells in the apical cytoplasm, extending all the way through the cell. BEST4 is in a reticular pattern in the basal portion of the cell, suggesting that it traffics also in the gut. Nothing is really understood about bestrophin 4 in the intestine. So because these cells are absent in the mice, we developed a rat intestinal organoid model to be able to study these cells, and in collaboration with the Sumigray lab. And as you can see in the differentiated organoids, several of these T cells can be observed, but not in the undifferentiated organoids. And they can be marked by CFTR in mice 1 and bestrophin 4. So we do have a model to study the cells now. So we looked at OTOP2 in vivo in the rat jejunum, where these cells are most abundant under acidic conditions. And here you can see when we use neutral pH, there is no OTOP2 staining in any epithelial cells in the villi, but the mice one is detected. But when you lower the pH, there's rapid upregulation of OTOP2 staining in the apical domain of these cells. Now, the folks who looked at BEST4 positive cells in the colon published a paper using rhodo-red fluorescent dye, which allows demonstration of proton import into the cell. And here they showed in that study that when you use rhodo-red, there's maximal uptake of protons at pH 5, which is consistent with the CF intestine. We just completed some studies in our rat organoid model, and we have been able to reproduce this, confirming that the Chi cells in the intestine are also picking up protons. So what about CFTR and GCC? We looked at that under conditions of acidity. And here you can see that CFTR also traffics rapidly when the pH is lowered in the lumen, as shown by the fluorescence intensity. GCC is doing the same thing. Now, GCC is present in the apical domain in the villus enterocytes, but enriched in Chi cells also, and it robustly traffics to the apical domain under acidic conditions. The lumen of the intestine is able to buffer that acidity, so the pH is restored when you lower the pH pretty quickly, indicating that these cells, in addition to other cells, are able to deal with the acidity through buffering capacity. So I'll switch gears and talk about neuropod cells, because the EECs are cells which sense luminal contents, and these cells are known to secrete hormones both locally and distally through the bloodstream where they act. But more recent studies have been able to identify these basal processes in EECs that extend into the lamina propria, and these basal processes have synaptic vesicles which allow rapid transmission through afferent and efferent neuronal interactions with the ENS to respond to luminal contents. So these neuropods contain a lot of different components to allow this rapid transmission. And we looked back at our single cell data, and indeed we found a number of genes that are present in neuropod cells, including tubulin beta 3, which is a neuronal marker for the ENS. The GLP2 receptor was found also in TUB3 positive neurons. Synaptotagmin 3, which is involved in synaptic transmission. Neurofilament medium, which is a subunit of neurofilament protein in neurons and abundant in EECs. S100A6, which is a calcium binding receptor in neurons, also important in cell differentiation. CHAT, which is the enzyme that produces the neurotransmitter acetylcholine, and of course GCC. A recent study in 2023 identified neuropod cells in the mouse small intestine enriched in GCC, and those cells have been shown to be important in cyclic GMP-mediated visceral hypersensitivity that's important in IBS. So we looked at our proteins in the Chi cells based on the genes. And here you can see TUB3 positive cells in close opposition with BES4 positive cells in the lamina propria. Similarly, synaptotagmin 3 in yellow in close opposition to the BES4 positive cell. The neurofilament medium, we find it in the Chi cells but at the base, but also in all epithelial cells on the villus epithelium, indicating that there is some unrecognized previously crosstalk potentially between villus cells and the neuronal system. S100A6 is enriched in the apical domain of these cells, as shown in green. And chat-positive neurons are found in goblet cells, as expected, because they're important in mucus secretion, and in the Chi cells shown in yellow. And we always see these Chi cells in close proximity to goblet cells. So the GCC, as I mentioned before, is enriched both in the apical domain and throughout the cytoplasm of these cells. You can see by 3D reconstruction the location and the proximity of the TUB3 positive neurons with a BES4 positive cell. Oh, where's the mouse? I want to be able to show this video. And this is a video. Anyway, so the relevance of these cells to CF is really unknown because the lack of these cells in mice. So we were fortunate to get rat tissues from our collaborators in Adelaide. These rats were generated with the DF508 mutation, and also we got knockout rats. And we used the tissues to do in situ hybridization studies looking at CFTR and GUCA2B. And here you can see very nice RNA in the wild type, in the villi, and in the superficial crypt. And GUCA2B is preserved in the wild type, both in the villi and in the superficial crypt. In the delta F508 animals, there's marked reduction of RNA, both on the villi and the superficial crypt, but GUCA2B is preserved both on the villi and the superficial crypt. And what about CFTR and GCC? We looked at that. And if you look at the wild type tissues, very, very nice staining for the cheese with mice 1, but in the delta F508, marked reduction of CFTR staining. But in the knockout animals, mice 1 is preserved, but there's no CFTR, indicating that CFTR is not important for the specification of these cells. The GCC is preserved, but seems to be upregulated in the delta F508, both in the epithelium and in the high expressor cells. And as you can see by the fluorescence intensity, it's markedly increased. We also noted a 3-fold increase in the abundance of these cells in the delta F508 animals, indicating that there is some compensatory response, as John indicated, in the tissue. So in conclusions, what I've shared with you is that what we're finding is that these Chi cells in the intestine are neuropod cells. They appear to sense and respond to changes in luminal pH in the proximal small intestine, and it suggests that these cells may play a significant role in CF pathogenesis in the gut. There's a lot of work to be done to understand the role of these cells as neuropod cells, the role of BES4, and the role of these cells in human CF gut disease. The majority of the current work was done by a postgraduate student in my lab, Jason Jin, together with other members of the lab. We have an active collaboration with the Summergreen lab at Yale to look at the cell specification. And I'm grateful to my colleagues at the University of Adelaide for the rat tissues, without which we couldn't show the importance in CF, and our grant supports from the NIH, CFF, and the CFRI. So I'm happy to take any questions.
Thank you. Any questions from the audience? John.
It's interesting that the Chi is derived from a common progenitor of the neuroendocrine. Is that what you said?
Yeah.
So, I mean, it's the same for ionocytes in the airway. So if you lineage trade, well, I guess mice sometimes we wouldn't know if they come from the cryptid.
That's our big challenge, John. It's a big challenge.
What's the current thinking? Are they going for some other non-epithelial population?
Well, BEST4 is not expressed in mice. And so we are looking at that now, trying to understand, why these cells are not in mice. But interestingly, as I mentioned to you, if you, I've made transgenic animals, knocking out other genes that I've been studying. And when you perturb genes that also regulate CFTR in the gut, we see these cells in the intestine, and I published that. So, for example, I made an animal model of microvillus inclusion disease, which causes severe diarrhea. And when we knock out myosin 5B, we see these cells in the gut. And in another mouse model I made for another gene that I'm studying, we also see them. We haven't completely characterized them like we have in the rat, but we see them. So we need to understand more. The problem that we have now is because they're not in mice and because BEST4 is not expressed in mice, it's a big challenge. That's why we made this rat organoid model so we could try to study them. We've knocked down BEST4 in the rat organoids, and when we knock out BEST4, we do see changes in fluid accumulation on the 3D level. But we think these cells have some important role in the acidity issue. We also think that these cells are sharing some features in terms of their development with EEC cells because they're expressing very high levels of uroguanilin, right? And why is that? Interestingly enough, when we look at the GUGA2B positive cells and uroguanilin expressing cells, cheese express uroguanilin and GCC and GUGA2B, but not all GUGA2B positive cells are cheese. So it gets more complicated.
Any other questions? Thank you so much.
Great, thank you.
So before wrapping up this workshop, so we have 3 questions. 1 question for Celeste and 2 questions for Sharon. So first question for Celeste is, because you see enhanced degradation in delta 508, would you expect to see similar degradation with other rare non-delta delta 508 mutations, and also that treated with read-through NMD plus correctors from Dr. Alexander Stauffer?
Okay, can I answer?
Yeah, yes, please.
I don't think it's on, so
You can come up. Yeah.
it's easier.
Sorry, that was a bit of a long question, so I hope I will answer what I can. So we currently are trying to look at a lot of different mutations with this system, particularly those that are corrector resistant, so they don't respond to corrector drugs. And we've looked a bit into N1303K, and yes, we do see, quick degradation in our cell model. I'm sorry, are there any other?
And also the rare mutation treated with read-through NMD plus correctors.
I have not looked at NMD correctors. I have looked at a variety of different correctors like tezacaftor, Chlor4A, and elexacaftor. And really what we're seeing is that the quality control pathways are dependent on the subcellular localization rather than the specific corrector type.
Thank you. The other questions are for Shaolong. Actually, relevant question to the previous one. So, do you plan to study CFTR inhibitor 172 binding or interactions with CFTR peptide derived from other non-delta 508 mutations?
Good.
Can I see the question?
Yes.
Which one?
This one.
This one.
Yeah.
So, do you plan to study CFTR binding or CFTR peptide derived
From rare mutation.
Host treatment is very different, such as.
It's basically on the effect of
So,
inhibitor one.
honestly, we don't have
You should repeat the question for the people in the room. Yeah, so that's a.
It's kind of a long. So.
No, I can kind of just.
The question is, do you plan to study CFTR inhibitor 172 binding interactions with CFTR peptide derived from PTC variants post-treatment with reduced-role NMD compounds such as G542X, R1162X, W1282X? Would you expect to see differences among variants? Well, honestly, the answer is we so far don't have a plan to do this. Maybe this will be something in the future. It's the short version of this answer to this question.
Yeah.
Is also the second one?
Yes, second question.
So
Thank you.
the second question is, when I say lentivirus are a good system to express CFTR, is it true in any cell context, or the nature of the cell transduced with the lentivirus could affect its folding? So from what I know, this system works with both CHO cell and HECA cells. So the samples we got is from CHO cells. So it works very well with the CHO cell system. I'm pretty sure when it was first developed, they tried the HECA cell as well. So I think it should work with that too. So that's the answer to the question. I think I answered the question.
Yes.
Yeah.
Thank you.
Okay.
Thank you very much.
Okay, so we are almost. It's almost time. Thanks so much for joining this workshop, and have a great time at the rest of the conference. Thanks so much.